Okay, so we're finished with the first half of Biochem 2, which kind of dealt with carbohydrate metabolism with glycolysis and glycogen metabolism and the citric acid cycle, electron transport and oxidative phosphorylation, and we kind of even threw photosynthesis in there in terms of our first kind of anabolic pathway too, to think about the synthesis of carbohydrates in plants. So now for the next few chapters, and to wrap up the course, in Chapter 20 we're going to deal with lipid metabolism. Chapter 21 will deal with amino acid metabolism and protein metabolism. We'll kind of skip ahead to Chapter 23 where we'll do nucleotide metabolism, and then we'll come back to Chapter 22 and just kind of finish up with kind of this global view of metabolism. So we're going to take 4 lectures to cover lipid metabolism. We spent a long time covering carbohydrate metabolism, weeks in several chapters, but we're going to spend 4 lectures, and we're going to deal with first thinking about how we actually get lipids into our cells so we can actually do some fuel metabolism with them. So we have to think about digestion, absorption, and specifically for lipids, because they're hydrophobic molecules, we have to think about transport. So that'll be our first lecture. Our next lecture will deal with the breakdown of fatty acid molecules. We call this fatty acid oxidation or beta oxidation. We'll also talk about a water-soluble form of lipids called ketone bodies. Our third lecture will deal with fatty acid synthesis, and so we'll kind of think about the anabolic process of synthesizing lipids for storage. And then our last lecture, we'll talk about regulation, talk about the synthesis of other lipids, we'll talk about regulating cholesterol metabolism. We may even kind of dip into a 5th lecture as we maybe try to do a case study in class. So again, thinking about carbohydrates versus fats, we've been very carbohydrate focused, and that's because that's kind of the body's preferred fuel, specifically in the brain and red blood cells. So again, several chapters dealing with how we metabolize carbohydrates. What we're going to move on to now is talk about fatty acids, and again, that's nature's most efficient form of stored energy. We know this already, right? Carbohydrates are worth 4 calories per gram, fats are worth 9, so a lot more energy in a lot less space means we're going to use that for long-term storage. And really we have unlimited fat reserves within our body. We have limited carbohydrate reserves, right, about a half a day supply of glycogen, but fat reserves are unlimited in terms of their capacity. So we'll do a little review in our activities on the structure of dietary fats. Chapter 9 was a long time ago in Biochem 1. So thinking about the structures of triglycerols and how that relates to how we need to think about transport issues, and then also thinking about the breakdown of these species. This is going to be a figure we'll refer to a lot in our activity because it's kind of a nice picture that talks about both exogenous or external dietary sources of lipids and then endogenous sources of lipids. And again, those are going to be ones that largely come from our adipose tissue. So we'll spend a good bit of time dealing with just how do we get these species into our body. And talking about exogenous first, we just have to think about how we get these water insoluble species out of our digestive tract and into our bloodstream. And remember, this is a big picture theme. Whether it's carbohydrates, whether it's lipids, whether it's proteins, we do not take those kind of polymeric forms and take them from our digestive tract and get them into our bloodstream in that way. Only monomeric forms, so free sugars, simple sugars, so monomeric sugars, amino acids, or in this case, fatty acids. So what we need to do is we need to take that triglyceride. Remember, this is our triglyceride. We've got our 3 carbons, and then we've got our fatty acids appended from that. We need to snip these fatty acids off, and we've got enzymes that will help do that. But unfortunately, those enzymes are water soluble, and this guy is not. So within the stomach, we've got emulsifying agents like bile acids, and then we've got this churning peristalsis that's going to kind of mix our oil and water layers, and that's going to allow these enzymes to access the interface of these oily molecules and snip off these fatty acids. So we'll spend a good bit of time talking about that in class as well. Again, these bile acids are very important. They're synthesized in the liver, stored in the gallbladder, and again related to cholesterol. So that'll become important as we deal with cholesterol metabolism. So again, we're not done with reaction mechanisms. We'll deal with hydrolysis of an ester. So again, triglycerides, we've got a triester that we have here to think about hydrolysis of. So, and we'll think about how these enzymes, again, get at these fatty acid micelle structures to be able to do this chemistry. All right, so we've snipped off fatty acids, but again, fatty acids are not horribly soluble in water. So once inside intestinal cells, we actually have to complex them with proteins, and this is kind of a theme that we'll see for these molecules from this point on. And there's a protein called a fatty acid binding protein that's within your intestinal cells. Big picture here, most of the absorption of fuels and metabolites that we've consumed from our diet happens in our small intestine. So we have these fatty acids that have been snipped off of our triglycerides. We get them into our intestinal cells, and to increase their solubility and also kind of help hide them so we can pull more fatty acids in from our digestive tract, we complex them with these fatty acid binding proteins. Once we've got them into our intestinal cells, we're actually going to repackage them as triglycerides, and then we're going to complex them with other proteins to facilitate their water or their solubility in our bloodstream. And so you've probably heard of things like LDL and HDL. Well, we're going to talk about some other particles, and I apologize, this is a pretty poor picture. It was just one I was able to find online that I think does a good job of showing the difference in size between what we call chylomicrons, and we'll talk about what chylomicrons are, and then particles like HDL and LDL. So we'll learn about these different particles here. And again, this exogenous pathway is how we take dietary fats, these triglycerides that we consume in our diet, we emulsify them in the stomach, we hydrolyze into free fatty acids, and in the small intestine they're taken up by the intestinal cells, complexed with our fatty acid binding proteins, repackaged as triglycerides, and then also complexed with cholesterol, and made into these kind of giant water-soluble lipoproteins that we can kind of transport in our bloodstream. So kind of the first exit of these are particles that we call chylomicrons. And this is kind of another nice picture that shows the massive size of these chylomicron particles, and that's just because they are chock full of cholesterol and triglycerides. We'll spend a lot of time talking about LDL here, but I like to think about these as kind of being this overloaded bus, and that's going to help us think about why LDL is bad. You know, they talk about good cholesterol versus bad cholesterol. But ultimately, at the end of the day, what's the difference between HDL and LDL are the proteins that are on the surface. And really we can think about those proteins as being directions for where these particles go. And it has to do, and that's again what makes them good versus bad. LDL particles have proteins on their surfaces that direct them from the liver to the peripheral tissues. For HDL, a different set of particles is actually going to direct them in the opposite way, from the peripheral tissues to the liver. And that fundamentally is what makes HDL the good cholesterol. So again, cholesterol gets a bad rap, but remember, cholesterol is just, you know, a four-fused ring steroid-based molecule. What it's complexed with is really what makes it good or bad, and fundamentally at the end of the day, whether or not it's kind of deposited in your cardiovascular system where it doesn't belong is truly what makes it good or bad. So we'll spend a good bit of time talking about the different kinds of proteins that are present on the surface of these lipoprotein particles. And again, that's going to direct where these guys go. So again, what we're going to spend a good bit of time is kind of dissecting this picture, making sure that we understand that once things are absorbed from the intestinal tract, their first stop is really the liver. And so we're going to talk about how the liver then can repackage these and send these out as VLDL and particles, which as they offload their contents become smaller and smaller, so they eventually become these low density lipoproteins. So we'll pick apart a lot of that in our activity. I don't want to spend too much time going through the details there, but again, peripheral tissues that we'll spend time talking about are muscle cells, and these two different colored arrows here remind us that muscle cells take up both exogenous and endogenous triglycerides. So again, just to remind ourselves that cholesterol is not good or bad, what it's packaged with and what it ends up doing in your cardiovascular system ultimately makes it good or bad. We'll spend a little bit of time with cholesterol, initially here talking about how you take in cholesterol from your bloodstream. And so the uptake of LDL particles is how cells get exogenous cholesterol. And so we'll kind of walk through this process, and we'll talk about how this process can kind of downregulate the endogenous synthesis of cholesterol. Because remember, cholesterol can be from your diet or you can make it yourself. So we'll talk about the make it yourself aspect of it at the very end of this chapter. But we'll talk about here how we uptake LDL particles to get that cholesterol. And we'll spend just a little bit of time, this will be something that we'll be focused on more with our last section on regulation, how we actually get lipids from our endogenous sources, from our adipose tissue. You'll notice that we don't have arrows really coming out of our adipose tissue here, so we'll deal with that more at the end of this chapter here. So at the end of the day, fat is going to become really important because it is a very valuable fuel source. And again, if your blood sugar is low and your glycogen's depleted, you're going to tap into those triglycerides as fuel. But remember, these cannot be made into glucose. So we'll talk about how we can break these down in our next chapter or in our next lecture. But that process again is called beta oxidation. And to just give you a little bit of preview, right, we've got all of these carbon atoms hooked together in essentially what's a hydrocarbon, and we snip them off 2 at a time, and we're going to see some chemistry that's very similar to stuff that we saw in the citric acid cycle. We're going to do reduction, hydrolysis, oxidation, hydrolysis, oxidation, and then cleavage. And so that series of reactions cycling around is going to be how we get these fats to be broken down. But we'll deal with that in our next lecture.